Mahalaya.
Yes, I'm sorry. Good morning and good afternoon, everyone. This is Mahalaya Marshall from the IQ Consortium Secretariat. On behalf of the IQ Consortium, I'd like to thank you for joining this morning's webinar entitled Time Course and Model Fitting of CYP Induction Data, Impact on Drug Interaction Risk Assessment. Today's presenter is Dr. Simon Wong. Before we begin, I'd like to take care of a few housekeeping items. First, all audience members will be on mute for the duration of the webinar. We ask that attendees submit questions throughout the webinar via the question module within the control panel located on the right-hand side of your screen. You may undock this module by clicking the small arrow in the upper right-hand corner for easier question entry. Attendees may experience intermittent delays in slide transition due to local bandwidth demands. Finally, slides and the recording of this session will be posted on IQ Connect and the IQ website at the conclusion of the webinar. Once again, thank you for joining us for this webinar today, and I'll turn things over to Naresh Hariprasad, who will introduce our speaker.
Thank you, Mahalaya. It is my pleasure to introduce Dr. Simon Wong to all of you. Simon is a director at Pliant Therapeutics, where he leads the drug metabolism and pharmacokinetics group, continued evaluation and optimization of ADME properties of preclinical candidates. Previously, he spent 3 years at Genentech and 12 years at Amgen supporting DMPK with an emphasis on DDI risk assessment and small molecule project support. He received his bachelor's in life sciences and PhD in pharmacology and toxicology at Queen's University in Kingston, Ontario, Canada. He has been an enthusiastic contributor to the Induction Working Group since 2016 and continues to be both perplexed and intrigued by the translation of in vitro models to describe and predict in vivo phenomena. Simon, over to you.
Hey, thanks, Naresh, for the nice introduction. It's a real thrill to be here. I couldn't help seeing everybody log in, and it's just great to see some old friends and different colleagues from a variety of different places. You know, this is probably the closest we can get to an ISSX or some kind of meeting. I wish I could just, walk down the hall and catch up with you guys. It's been a real thrill to be part of this induction working group, and it's kind of been a labor of love for me, spending countless weekends going over this data, and it's just a super thrill to be able to share this work with you guys this morning. And hope you got your coffee or your lunch ready. And I want to thank all the folks that have logged in. I know people are busy and your time is valuable, so thanks for lending me your ear for the next hour or so. Just want to check everyone can hear me. You can't really speak. Sorry, you can type. We're monitoring you. We can see your texts come in, your questions come in. I can also tell if you're paying attention or not. I can tell if you're focused on the screen or not. So you keep that in mind. Let's just get into this. If you tuned in last week, you heard a little bit of the IQ, and I think there's kind of a certain amount of clout and mystery about this, just with the name. But I think if you think about the overall goals and the mission, it's quite a noble one in terms of trying to push innovation and science to the next level, and kind of emphasize a data-driven approach towards perhaps adjusting the way we think about regulatory requirements. And in this particular case, we're talking about drug-drug interactions and induction. And I wanted to add that just because of this industry, and it's large pharma, essentially, those are the main contributors to these IQ groups. You've got a lot of resources. You've got a lot of folks thinking about this with the time to generate a lot of data. And I wanted to emphasize that. I mean, you can read papers that kind of explored similar topics, but they don't have the resources and the amount of information to help back up the conclusions. So I think it's great. And when I kind of dropped into this during my time at Genentech, I was just kind of initially overwhelmed by the amount of information and data that we had to process and analyze. And I'm kind of a data junkie, and that's why if you look at the LinkedIn posting, I'm going to kind of geek out on that a little bit. Indulge me, and we'll kind of explore the way our path went through sorting out this data and coming up with some recommendations. If you, it's a little bit slow on the slides here, maybe on the next one. Here are the folks that are contributing to the work here. I bolded them. the authors on the actual manuscript and the paper. And shout out a few folks here. I think Naresh and Diane, you heard her speak last week. And I think it's just been a real thrill to be part of this team and to share this work. Don't want to dwell too much on these kind of slides showing you the structure, but I think what's obvious is we've had these subgroups, and over the past 4 years, almost every year, you can see we've put out a nice paper in DMD. You can go look those up, and I think there were references in the previous presentation. And you know what's a little bit sad about this? I'm not sure if you noticed this in Diane's presentation. It's the sunset. So this is actually the end of this working group, and I think it's the culmination of that work. And Diane summarized it last week. I'm going to try and cap it off here with the last data analysis on this time course business and this fitting the curves. So let's get into it. Okay, so I wanted to just kind of ease into this and show you kind of intuitively what we're trying to answer. And again, there was so much data when I joined. I wasn't really sure how to approach the topic. And if you just think about time course for CYP induction and what's going to happen over time, I thought this is a nice way to introduce you to that. So if you look at time on the x-axis and fold induction, and this is just a single concentration of the inducer. I'm just showing you 1A2 or 3A4 with omeprazole or rifampin, mRNA activity, and just qualitatively, what do we see here? And we're seeing, well, let's start with the big thing. We're seeing variability. Each of those lines is a different lab generating that data. So there should be seven lines in there. And this is, again, this is just one high concentration. And you can kind of see the trends we're trying to get at here, right? You're seeing at 6 hours, not much fold induction. Let's just look at mRNA. And then you're seeing, I'm focusing actually on the omeprazole 192 panel there. I think I should turn on my little laser pointer so you can see what I'm pointing at. I'm just focusing on this because this is a nice kind of classical. If you just came off the street and I told you we're looking at CYP induction in time, you'd probably think it looks like this. And the questions we're trying to answer is, so, okay, you've got 48 hours, 72 hours. If you're up on the guidance, you know that the FDA kind of waivers and says, yeah, you can use 48 or 72-hour incubations. The EMA is a little more hard on that. You should use 72 hours. So if you ask me, what is the key super high-level take-home message? Is like, well, what's the deal with that? Can you get away with 48 hours? And you can see, I'm just looking at this one again, you can see the kind of trends we get out of that. And you also can see the variability in the different donors, which is well documented. If you look at Jane Kenny's paper from 2018, you can actually see some somewhat scary numbers in terms of the variability. I'll just throw some out here. 130-fold range in the EC50 for CYP3A4 mRNA. And this is, again, well documented if you know about this. But if you kind of not super familiar with CYP induction, that's a staggering amount of variability, right? And just think about mRNA and basal levels and what's happening in cell culture. You can appreciate what's going on there. And the other kind of mind-blowing stat is, and again, this has been in the previous papers, up to a 10-fold range in the EC50 from the same lab, from the same donor. So think about that. So just context all this information I'm trying to show you with that kind of variability. How are you going to get your head around showing a difference between 48, 72 hours, or is there a difference or not, when it's kind of lost in that variability? So let's kind of step through how we approach that. Again, I'm just showing you these slides that can kind of ease you into it, and then trying to introduce you to the inducers and the CYPs that we investigated. So let's take a look at phenobarbital, again, mRNA. You can see that for some of these donors, it kind of went up high and stayed high, and it didn't really change. And I think that's an interesting observation for message. But if you look at activity, we know, and we see this is kind of a theme I want you to pay attention to. We see this, and I didn't really mention it for these two isoforms, but you can see activity. You see what you would expect a little bit more in terms of it gets, the fold induction increases more obviously with time. But with mRNA, you're seeing that in some cases it's already peaking and kind of high for the entire time, at least 24, 48, 72. So this is kind of fun to look at qualitatively, but this isn't really good enough, right, to get to any strong recommendations. So let's look at this more from a perspective of traditional curves and rifampin and 3A4. You can see here, this is intuitively what you might think would happen, right? You would fit these curves, estimated Emax, EC50, and you can see what it looks like at different time points, message and activity. Now, when I look at this, I would say, all right, looks like mRNA, the greens and the orange and the purple, so 24, 48, 72, they all kind of overlap. So that looks like those parameters look about the same, right? I'm not showing you the SD there because that's a scary number. And if you look at the activity side, okay, so we talked about this all overlapping, you can see there's more separation here, right? And maybe that's more obvious. It's more obvious to me because I've been staring at these things for 2 or 3 years, but you can see the separation here a little bit more. So you might already think that message is going to be a little bit better in terms of correlating with 72 hours than activity. But let's try and figure out a better way of trying to understand that agreement. So now I'm just plotting the Emax by mRNA or Emax by activity with time on the x-axis, and you can now a little appreciate more of the variability we're seeing here. So when I first joined this, I thought it was just a simple ANOVA question or a stat question of showing statistical difference between 48 and 72 or lack thereof, and you can declare there's no difference in fold induction, and you can make a conclusion based off that. You can see you can't do that with this kind of variability that we see across the donors and the labs. You can't, these are all not statistically different. Now you can qualitatively say activity, yeah, I see this increase, but you're not going to get anywhere there. So if you think about, again, I'm just showing you more pictures of how this looks. And now what I'm plotting here are box plots of the Emax with all the inducer isoform pairs. And now you can see at 12 hours, this is the kind of range. And look at that, that full difference between the different donors. Can't really quantitatively understand that agreement there. I see. Yes, these slides will be available. Don't worry. Okay, I can't be distracted by these questions. So now you can look at activity, and just as I said a little bit earlier, you can see a little bit better separation, especially with the CYP3A4. Now, when you think about agreement, a traditional way, if you think about, let's say you're looking at observed versus predicted, the classical question for DNPK scientists, you probably will see something like a correlation plot, which we're showing here. So let's assume for a second 72 hours is where you want to be. Now we're comparing each of the earlier time points to 72 hours. And I think you've all seen plots like this. You're trying to optimize your number of points within twofold. Those are the 2 lines, and you conclude that there's a good correlation. You might throw up an R-squared. I think that's great. This is kind of drifting towards the conclusion we want to get to. But there's a better way. And I don't want to go into too much detail. Some of you may have heard me go on about this Vlad-Altman plot. Go take a look at it. Here we've got a little bit better quantitative way of understanding the agreement between the earlier time points and the later time point. And again, we're assuming that 72 hours is the kind of gold standard here. And we're looking at the fold difference between the measurements at the earlier time points compared to 72. And now this is kind of that correlation plot stuck on its side, right? You're now looking within twofold here. And you can see, obviously, at 6 hours, you got an under prediction, as you would expect, both message and activity of what you're getting at 72 hours. And this is getting more quantitative, right? If you read those papers, we're plotting it actually against the x-axis, which is the average between the 2 measurements. There's ways to interpret that and what that means, but eventually we thought that that's not relevant to our question. We just want to know this average fold error between the measurements at early time points to the late ones. And let's just cut to the chase now. We use this average fold error approach. Now this essentially just compares the measurement of the parameter Emax EC50 at the early time points compared to 72 hours and allows us to kind of quickly visualize the differences. Now you can see here, obviously at 6 hours, majority of these points is below the twofold, so you're under predicting. But the key message is that at 48 hours in particular, both message and activity, you're seeing this excellent correlation. That's the wrong word to use, a low fold, average fold error, 0.95, 0.87. So you're seeing that excellent agreement, that's what we're calling it, between the two measurements. And this agreement, if we want to define what's good is within twofold, you're seeing that agreement is there at 24, 48, and maybe even 12 hours. So let's take a little dive a little bit deeper into, well, what about the different isoforms? So what I was showing you here is everything. But let's just take a peek, and I don't want to dwell too much on this because it's not super relevant, but it's interesting. And so again, you want to be an average fold error within twofold. We define that as a good agreement. And now I'm showing you what it is at each time point for the different CYPs. Now there is no green line over here because we didn't evaluate CYP2C9 activity because of the lack of robust substrates or response. No, totally other topic to talk about. But what we can see here is that, yes, at 48, 24, they're all within twofold for message. And okay, 48 looks pretty good for activity. 24, you know, you're kind of bumping in within a, not quite within twofold for activity. And what I thought was interesting, if you kind of look at the breakdown in CYP isoforms, is 1A2 not doing as well as, say, 3A4. And you can see at 12 hours here, the fold error for the 1A2 is indeed less, it's greater, right? So it's not within twofold compared to 3A4. Now, this is something you can think about and pontificate, don't want to right now, but I just thought I'd point that out to you. The key message, though, is that the 24 and 48 hours in terms of message, and maybe even 12, right, are within twofold. Now, if you want to crystallize the key message about the time course, this is the table that you look at. And essentially it's reporting the average fold error for the Emax EC50 and then the Emax over EC50, comparing these early time points, again, to the 72-hour assessment. And I think the key message is, as I've been trying to tell you, excellent agreement between 48 hours and 72 hours for message as well as activity. Now the question is, can you push it, right? How comfortable are you using earlier time points? And as I showed you in the plots, 24 hours for message looked pretty good. For activity, a little bit outside the twofold comfort. So we don't recommend activity for 24 hours. But for message, we feel that it's within twofold and offers you a robust response. So if the Emax and EC50 actually correlate, I think it's pretty obvious that if you throw them into an equation, they would also correlate. But this is just showing you the actual risk assessment. So if you take your Emax and EC50 determined at those earlier time points and throw them into your AUC-R equation, which I put there, let's see what happens to your predicted AUC ratio. Now, I hope you understand this is an induction, right? So you're getting a fold reduction in AUC, so that's why you're going down on the y-axis. And you can see 72 hours, this is the prediction we get, and then the prediction you get at the different time points based on those Emax and EC50s. Don't want to dwell on this, this is telling you the same exact thing. The 24 and 48 hours for message are doing a pretty good job of predicting the actual risk assessment you would have done at 72 hours if you had measured your Emax and EC50 at those time points at 72 hours, and you can see the average fold error there. And then same actual, a little bit maybe more obvious now for activity, when you go ahead and use that equation, you can see the 72-hour and then 48-hour correlation and the average fold error there in a good agreement. And then of course, 12 and 24-hour activity, you know, you underestimate that AUC-R by more than twofold. I put this in there, and I kind of wanted to advance to the data fitting. So I'm not going to talk too much about this. You can read the manuscript about it, but it's a very simple kind of extrapolation of what we said in the previous manuscript, which is how do you define a positive or negative CYP inducer? And there's a lot of stats behind that. But the take-home message is if you see concentration dependence and twofold induction, and you also see your rifampin response at greater than 10-fold, you can classify that as your positive for induction. So, okay, well, what about earlier time points? And you know, this is a kind of complex table. Let's just focus on the bottom row here. Well, rifampin actually at earlier time points did in fact give greater than 10-fold response for quite a few of the donors tested. And so that recommendation there is, well, if you're just using that criteria, and the same logic applies, right? You could use an earlier time point to assess positive for CYP induction if it also follows those or satisfies those criteria, greater than twofold and greater than 10-fold for your rifampin. So here's the key recommendations on the time course. And I feel like I'm being a bit repetitive, but I kind of wanted to walk you through how we got to it using the average fold error approach, and a lot of data that was used to support that. And it's essentially that the average fold error is within twofold for 48 hours, even down to 24 hours. And then the last thing I mentioned was the twofold criteria for positive for induction if your rifampin response is decent. And that kind of caps off the time course part of the presentation. I think kind of right on time. Let me see if there's any questions. Nothing I want to answer right now. I'm going to go on to the fitting part. Now, kind of to introduce the fitting part, again, when I dropped in onto this project, one of the first things that came out was, hey, are we using a consistent approach on fitting these curves? And that's particularly relevant for this work because there's some crappy curves here, especially at the earlier time points. And so imagine walking into a room, we had this data jam and tables and tables of Emaxs and EC50s fit by the individual labs using their own individual rules, and they were inconsistent. And so we realized right then that, oh, this is not going to work. So what I'm showing you here is actually the full difference between, this is Emax estimates, and each of those numbers is a different lab and the different CYP inducers and the CYP that they induce. And what we noticed eventually was for curves that are kind of not reaching the Emax, so this curve here, which is probably a very commonly encountered scenario, depending on your rules and how you fit that, you're going to get a different parameter estimate. And so that needed addressing. Now, if you have a nice, well-behaved curve, like a rifampin, and you define your concentration range to define the Emax and EC50, it looked like pretty good. Most of the estimates in the companies were getting within one fold, so that was reassuring. So based on this, we decided to devote some time to answer some questions. I'll just go through this quickly. So when do you actually start? You should start fitting. This is kind of an obvious one, but is there a true dose response? Should you start bothering to fit? Then the other more interesting questions is how do you address these weird curves? I haven't told you what a bell shape is, but you can probably figure it out. The induction kind of goes up and then starts dropping down like a bell. What do you do with that? Or what if you can't define the Emax? And is there a standard way to understand that or deal with that? Also, as you can imagine, for a curve that kind of looks like this, and your Emax estimate is up in the sky here, what Emax are you going to accept? What's a reasonable one? So we thought we should try and address that. And the other question was, so there are different flavors of nonlinear regression models that people were using. What does that impact the actual estimates? And then what's an overall recommended approach for fitting the incomplete curves or just curves in general? Now, I want to emphasize these are just recommendations, and some of them are somewhat qualitative, right? You can have a wide data set that we're looking at, and it's hard to argue, what is good, what is bad, but let me try and convince you based on what we have and then the breadth of data. The first one is a minor one, right? You just want to understand if there's a positive slope by Spearman's non-parametric correlation or simple linear regression. Is your response increasing with concentration? And that's kind of the first step. Most people obviously will know just by looking at it if you response or not, but we wanted to have a set criteria here on when to start model fitting. So let's get to the bell-shaped curve. This is actual data. I think this is an Faverin's curve with 3A4. Now, I think when some folks look at this, you probably say, oh, come on, the cells are dying here. You can just exclude those, right? Yeah, if your cytotoxicity assay is sensitive enough to test that or to actually detect it. I should tell you, we picked ranges of concentrations that we were known to be viable, that were known to be not cytotoxic. And so then we're still seeing that decrease. This recommendation that we're going to talk about is, well, you don't have any other reason to exclude those. So what kind of statistics you can apply to try and rationalize not using those as a curve? And I think we talked about this with the reviewers. Cytotoxicity assays are kind of gross measurements of cell death. By the time that happens, you're in pretty bad shape. And the general hypothesis is there could be a decrease in fold induction, something messing up your induction pathway, but the cells aren't dead yet. And so that could explain why, or down regulation. There's other excuses as to why it'd be going down. You don't have a reason to. So let's go into how you could address that. So to address, trying to remove points, the first thing we had to do was understand the variability in the triplicates overall. And I don't want to dwell too much about this, but it was about 30% CV. And if you know and understand that, you can use that to help guide what you feel is within an acceptable range if you apply a confidence level. So in this example, we're using a 0.95 confidence level. Our percent CV is 30%, as I mentioned. And it just so happens that gives you something that's somewhat easy to remember. The acceptable range is plus or minus 30% of the max fold induction. So now we've got a band of acceptance of terms of a maximum fold Emax we might accept, and also where we could throw out points where it appears to go down. And that's what this is showing you more explicitly. You've got this range based on the 95%, it's two-sided confidence interval, and the 30% CV, which we saw. Now, in the manuscript, we emphasize your estimate of CV might be different, your confidence level might be different. Just keep that in mind. So now we've got a somewhat easy way to remember. Now, I'll mention this. Some labs, again, in the working group, thought it should be 150%, right? So there's a comfort level there, and it's somewhat qualitative, right? What's your confidence level on a fitted value? But we put this one up because, well, honestly, I think it's easy to remember, and it's somewhat consistent with what people are doing in general. So the next thing we want to talk about today is okay. So what about all these different models? And I don't know if people are actually using these, but this is one of the member companies was in fact doing this. You can fit your data using a variety of different curves, model fits, and you can use your AICC criteria, something easy to do in GraphPad Prism, and it will tell you which is the best model. And you should use that one for estimating your Emax and EC50, right? There might be some statisticians on the call that are cringing on that approach, but this is what people do, and it's easy to do. So the question was, does that affect your Emax or EC50 parameter estimates? Now, and I'm not a mathematician, but if I look at these equations, I would think, well, maybe it would. Let's see if it did. So this is rifampin data, and this is, again, we picked concentrations of rifampin that are going to define that curve. So I would call this a well-behaved curve. Your plateau concentrations, your plateau Emax is actually well defined. And take a look at this. The 5 different model fits that we looked at gave essentially identical Emax or EC50s. Now, again, I don't want to dwell too much about this, but I got a kick out of this. If you use your AICC criteria, remember you want the lowest one. The lowest one is the 4 parameter. If you convert that to AICC weight, it's got the 94% chance of being the best one. And then you would, in your normal procedure, you would pick that one, right? Well, I don't think the Emax 3C50s, but the other ones, which are, quote, worse by a lot, are any different. And so this is consistent with what we saw, right? In the majority of the curves, you got the same Emax or EC50 values regardless of the fit you used, and that's because it well, I think the well-defined curves have something to do with it. So let's take that with a grain of salt. Then, if you've got a nice dose response, your AIC criteria may offer a limited value. We actually went back and looked at all the curves, and just to kind of emphasize that point, and I don't want to go too much into this, but essentially within 20%, and that's a pretty tight criteria, you're getting very similar Emax and EC50 estimates, despite the curve fitting technique that you used. So that seems great for rifampin. Now I threw these in there. You look at these in more detail later if you want. I think there is some question on, well, okay, when do I throw out an estimate? And I've mentioned for Emax, if it's above 130% of the observed Emax, you probably wouldn't trust it. But what about the actual model estimates? I think some kind of nice guidelines to keep in mind is keep an eye on that standard error, and you want it to be less than 50% of the model estimate. There's some rationale here. Just some general rules of thumb that you might want to incorporate into your data fitting paradigm to understand if your parameters are good or not. But it really depends on how well your data, your concentrations, your dose response approximates the classical curve. Now let's take a look at what happens for an incomplete curve. And this is kind of was a little bit mind-blowing part of the whole process for me, and in that they look quite different. And I had many conversations with folks at the Gordon Conference about this. And I think a lot of folks pretty much said, look, if you don't have data up here, you have no idea what that curve looks like. And I think that is the fundamental point here. I've seen this for candidates that we're trying to nominate. What do you do when you don't really characterize that full curve? And you kind of pragmatically say, now, at least the cases that I've seen when this happens, you're bumping up to your maximum concentration you can test in your in vitro assay, which reminds me of a philosophical debate which I've had with many folks. What concentration range should you really test in an in vitro assay? And my philosophy was always, well, you want to go the highest because you want the best characterization of the parameters you can get. And so go as high as you can in solubility or tolerability or whatever. And so people would say, yeah, but what about your anticipated therapeutic Cmax? Don't you want to be, within some kind of range of that? And I think, yes, I think you need to keep that in mind. And so if you use my approach and you kind of go as high as you can go, and maybe let's say this is 100 micromolar, right? You're sitting at well, what does that mean? And let's try and understand what that means if you were trying to estimate the true Emax for EC50. So we did the same approach here. Take a look at which is the best fit or not. And it turns out the sigmoidal three parameter fit in this case, according to the ICC criteria, is the best fit. Now, again, you might argue you still don't really know what that means. And so what you pragmatically do in this case, and when we talk to the folks as part of the consortium here, well, you just assume that your observed Emax is the max. So you force your curve to that, and you estimate your Emax as greater than that. Let's go to the next slide and we can look at that. And so if you take your regular logistic 3 parameter, you can see in blue, this is what it would predict. And again, that would be outside our acceptable range, so we would reject that value. So what you could do kind of pragmatically is, well, okay, I don't know what my true Emax is. I'll take my logistic 3 parameter or whatever. Actually, it doesn't really matter what model you have. You force it to your maximum observed, and then you estimate the EC50. Now you can see it goes from a blue to a red here. You would overestimate that potency, right? So it's conservative because you don't know the true curve, and you would kind of pseudo do your risk assessment based off that. Now, what was interesting, and this is one of the interesting things that came out of this thing, was the sigmoidal 3 parameter fit seems to estimate quite consistently when you fit it, the Emax of the observed Emax. So if you needed to actually use a model that approximated and made you, quote, feel better about the fit, you would use the sigmoidal 3 parameter fit, and it actually gives you the same kind of risk assessment as kind of the traditional approach of forcing a curve to the observed Emax. So that was a key recommendation there. Now, I didn't put this in the paper, but if you're following all along, I told you none of the models really matter, right? So if it's a good curve. But this curve, the sigmoidal 3 parameter fit, actually approximates the observed. So if you wanted to take a super take-home message, you could just use sigmoidal 3 parameter all the time, because it would cover the cases where you've got a good dose response. It would also conservatively estimate your Emax as the observed Emax, and then you would do your risk assessment assuming it's greater than that and greater than the EC50. All right. Again, I think I want to make sure there's enough time for questions. So you can go look at the guidance. I just want to remind you what this R3 equation was, and that there's a 10x factor on the Imax when you use it to predict the change in the exposure compared to the AUCR. It's going to come up important later. All right. Probably one of my favorite topics of this whole thing, let me geek out a little bit, is the initial slope approach. So we've talked about incomplete curves. We've talked about time course, but there's another toolbox up your sleeve that you can try. Now, intrinsic clearance, let's start with that. Intrinsic clearance, KMB max. I think everybody gets that. You can approximate that. When you do your substrate concentration super low, say you're 1 micromolar, you don't have to do your Michaelis-Menten anymore. You do that slope, you get your intrinsic clearance, it approximates Vmax over Km. Right? Everybody gets that, right? Not a lot of people know about this though. It's the same exact relationship for CYP induction. You're seeing the same kind of, classical Michaelis-Menten type curve, hyperbolic. You can, in fact, use that initial slope here to estimate the Emax over EC50. Just kind of common sense, right? Well, it was not common sense to me when I started with this. In fact, I didn't quite even understand what initial slope meant. I went and took a log curve and tried to draw a slope here. No, it's the linear concentration, it's the initial slope, it's the slope down here. Now, I've also got hill slope 1. What does this mean, right? Well, we did a sensitivity analysis to try to understand if you, in a perfect scenario, what does this relationship look like? And let's take a look at that. So this is just a zoom in of this, right? You want to be, this is only true if the substrate is way, way below the Km. This is only true if your concentrations you're evaluating is way, way below your EC50. So if you zoom in on here, now the only reason I'm doing this is to show the impact if your hill slope is not 1 and it actually deviates. So this is a simulation with the Emax is 100, EC50 is 2. So the Emax over EC50, the true value would be, well, be 50, right? Look at these slopes. The slope is 42 compared to 50. That's pretty good for when the hill slope is 1. But if the hill slope is not 1, you're seeing you're not really doing a good job of estimating that Emax over EC50. Now, if you have been paying attention, I hope you have, I could probably tell, you know that the 4-parameter or 3-parameter or any parameter fits all kind of look the same. And so in another analysis, we looked at, is the hill slope 1 or 2? And in general, it looks like the hill slope is 1 across the data sets that we looked at. So I think it's fair to assume that it's 1. Don't get into this space, you think it's 2 or not. So let's go with that assumption. Hill slope was 1. The initial slope will approximate it, at least theoretically, the Emax over EC50. Now, I don't want to emphasize this too much, but we've got a really a cool way now if you don't have a complete curve, right? But what you need, though, is data in this early part of the curve. So how much data? This table shows you, this is all simulation, though, a range of concentrations. And what if I just took that concentration and the fold induction I saw, calculated that slope? And how does that relate to the true Emax over EC50? Now, you can see here, this is the fold error. So basically the fold error on the slope versus the actual Emax over EC50. And you can see it's pretty good the lower your concentration is, right? The true EC50 is 2. And so you can think about what kind of concentration your range you need to really adequately characterize that. Now, keep in mind for this particular study, we didn't think about this going in. We picked our concentrations to cover the good range of defining the Emax. But nevertheless, we tried to see if it worked. So again, I'm showing you the same thing again, but let's just convert this curve, this incomplete curve, linearize it. Again, this is not log concentrations, linear concentrations. You can see our slope here is 1.4. Well, what was Emax over EC50? Well, that's the thing. We don't really know, do we? If it was a curve that looked like this, that would be the logistic 3-parameter, estimating its Emax of 39. The Emax over EC50 would be 2.2. Not too bad, right? This is a work in progress. I think it's super neat, but there needs to be more information, more data generated to understand the correlation. If you look at the entire data set, though, what happened? Well, let's just look at 48 and 72 hours. When we went into this, I had this kind of dream that maybe earlier time points would help. That's a distraction in this plot. Let's just look at, does that slope at 48 or 72 hours correlate to the true Emax over EC50 at 72 hours? Well, it's within 2-fold, but not the greatest, right? And I think it might be because we didn't design our experiment to do that. We didn't design it to stack those concentrations in the early part of the curve. And so that's one reason why additional work is needed to understand that. Philosophically speaking, I think some of you out there are crying out, well, Simon, you can't get that data because your response is too low at these lower concentrations. Your fold induction is going to be pretty wimpy. How can you get that? Well, yeah, that's the problem, isn't it? And maybe that's the next quantum leap. If I wanted to kind of pontificate just for a second, if our detection methods improve, if we can actually characterize the initial slope, maybe someday we don't even need to do your true Emax for EC50 and characterize the whole curve. Because we're doing that for intrinsic clearance, aren't we? We're measuring intrinsic clearance at 1 micromolar, a low concentration we assume is lower than the Km. That's what we're doing. Is that the next quantum leap here? Maybe. Something to think about. So I showed you that two-fold underprediction. What about the R3? And that's why I showed the R3 earlier. You chuck in this 10x correction factor for your Imax. Well, guess what happens? If your Emax over EC50 estimate using slope is twofold under, doesn't matter anymore. You got a 10x factor in there. So guess what? When you actually use the R3 using the initial slope, you get a pretty good prediction of the R3 if you use the Emax over EC50. So that's all just an artifact of the conservative nature of the R3 equation. You can afford to be twofold off with the slope if you chuck it into the R3 equation and you get a decent correlation with the average fold error being within pretty good of the actual predicted R3 using the Emax or EC50. Something to think about. I'm really trying to get this done with 15 minutes left, and I think I'm almost there. I'm just repeating myself now. But for well-defined curves, you can see that and I've said several times, like, all the models seem to do a decent job of estimating the Emax for EC50. But 8-point curves likely are not have enough concentration to get your initial slope, because we designed it this way to characterize the whole curve. We did not design it to characterize that part of the curve. Bell-shaped curves, we came up with what we called a statistically based reason to exclude these points that are seeing this paradoxical decrease in CYP induction above the past the Emax. That would convert that curve to an incomplete curve, probably the most common thing you're trying to deal with. And I hope you appreciate some of the solutions we've given you to deal with that. Using a sigmoidal 3-parameter fit, forcing your Emax to the observed Emax, or maybe using the initial slope to characterize that induction. This is the final slide in this presentation, but this walks you through the general flow scheme of how we recommend to fit the data, show that it's actually non-zero slope, go through the exercise of excluding points that may be part of that bell-shaped curve, fit with your data, your model of choice. It doesn't matter if it's got a decent dose response. And then the key one, which we've emphasized here, is what do you do with this if you don't reach the Emax? I just talked about all the fitting things you could try, but if you've been paying attention, you can use an earlier time point, right? If the cytotoxicity or whatever's screwing up your measurement at 40 to 72 hours, maybe you can get away with incubating for 24 hours. And you can get a more decent estimate of Emax for EC50, because we just showed you with the breadth of data that those parameters do correspond with the 72-hour measurement. I think I'm 2 minutes over. Look, I love talking about this stuff. Send me an email. We can talk more about this. We can follow up. You can ask a couple questions now. I think there's a couple questions on text that we can go through. Maybe I'll take a break and let Naresh ask those questions. Thanks a lot, guys.
Thank you, Simon. Really interesting talk. Entertaining too. Simon, maybe I'll start out with a question related to cytotoxicity, because that's something that I've been thinking about for a long time too. So for compounds that are cytotoxic and would possibly benefit from shorter incubation times, does the working group believe that 12-hour or even 6-hour can be used for internal decision making and induction?
Well, I think that's one of the kind of sticky points, right? Let's go back and take a look at, well, maybe even this one, right? So activity, definitely not. I think that's one clear thing we've been emphasizing. The average fold error at the estimates is just not good enough at 12 and 24 hours. For message, if your criterion agreement is within twofold, the answer is yes, you could use 12 or 24 hours and try and get an estimate again, the assumption is as the longer you incubate, the cells are dying, right? And they could tolerate a lower amount of time exposure to the drug. And so yes, internal decision making using earlier time points, I think, is something that should be on the table.
Yeah. Yeah, and I guess you want to use the right lots of hepatocytes as well, right? With that rifampicin response of about tenfold, if possible.
Yes.
Question from Albert Lee. He asks, which inducers routinely would produce incomplete induction dose response and in brackets non-saturating curves?
Right. That is a good question, and I probably revealed too much information here, but if you squint and look at these, at least the ones that we investigated, efavirenz 3A4 seemed to do it quite often, and you can see we're seeing these differences in the fold difference in the Emax estimate here. And so in terms of classical inducers, efavirenz did it. Interestingly, omeprazole 1A2 did it as well. So we're talking about curves that kind of look like this, right? That's the question, right, Naresh?
Yeah, he's incomplete, I guess.
Yeah.
Like the slow, where there's no Emax, essentially, no observed Emax.
Yeah. So either you can't test it because you can't get soluble, or the cells are dying, or whatever. You can't.
Yeah.
Yeah. And that was, I think, seen, as I mentioned, most commonly with these isoform inducer pairs. Efavirenz, I think, is the one we're most interested about.
Yeah, he asked the same question for bell-shaped curves as well.
Well, I think it's difficult, right? I think if you, and it was inconsistent, as this is why I'm kind of hesitant. We didn't see every lab's efavirenz 3A4 having two concentrations kind of crap out and look like a bell. It's really, so the variability that we see applies also to the tolerability that we see. In general scope, I think it's the same answer. It looked like efavirenz and the CYP1A2 omeprazole seem to be trending towards doing this this kind of bell-shaped approach. More specifically would be efavirenz.
Okay, thanks, Simon. Question from Dwipayan Mukherjee. Was there also high variability in the linear slope as the high variability in Emax? I don't know if that's, let's see. Is that, I'm trying to, was there also, so let me ask that again. Was there also high variability in the linear slope as the high variability in Emax? I guess similar to the high variability observed in Emax is what he was trying to ask.
Well, I have to be honest with you. It was hard to fit the linear part of the curve. In some points, there was 2 or 3 data points. And I think I said this a couple times. We didn't design the experiments to do that. And so it was pretty sketchy on predicting that initial slope. So yes, there's variability there. You can kind of see it here in this plot. I don't think it was. It's hard to comment if it was a lot worse than the actual variability in the fitted Emax, if that was the question. I think it's a matter of data integrity at these lower concentrations. Again, you're bumping against the sensitivity for your message, your mRNA.
Okay. Question from Dave, and he starts off by saying, very clear talk, Simon, so good job. How would the team differentiate concentrations approaching or defining Emax from those that is just making cells unhealthy and not responding as much as they should if they were healthy?
That's a great question, Dave. We need a better cytotoxicity assay, don't we? We need some kind of measurement of healthiness in terms of the induction pathway. What is that? I don't know. But I think that would really help us context these data, because we've gone in saying, yeah, cytotoxicity is at the end of the day. And by the time you see that, your stuff is totally or completely not valid in terms of this induction response. So I don't think there is one right now in terms of assessing that.
Yeah. All right, next question comes from Zhu Zhang. Besides cytotoxicity and solubility, any other reasons that could cause bell-shaped responses or curves?
Well, maybe Naresh, I'll call onto this one because I think you had the downregulation part of the paper in 2017, right? Is that the only other contributor?
Yeah, it's a tough one to answer. I guess it could be another contributor. There's also antagonism as well, right? I don't have any answers off the top of my head. Something that we could follow up with Zhu about later or after the webinar. Good question.
It is.
Let me see. Another question from me. With incomplete curves, would increasing the number of concentrations improve the accuracy of the predicted outcomes?
Yes, that's a good question, actually. So if you look at an incomplete curve like this one, right? The problem is, why is your curve incomplete? Because if you stack in, I think if you appreciate this, let's say I ran a whole bunch more curve points in here, that's not going to help you. If though you can kind of edge in more concentrations and tiptoe where you're not killing the cells or ablating the response, maybe, right? So that's the proper defining the Emax part. But it would definitely help you if you could have the sensitivity to go to the initial slope approach, right? So if you stack more concentrations at the lower end, yeah, and you can measure it and you get a better slope, sure, that'll help you. So I think it's a really good question. It depends where you're trying to bolster your dose response curve.
Right. Question from Helen Rollinson, one of my colleagues from AZ. In relation to cytotoxicity, did the team explore changes in housekeeping / normalizing gene levels as an alternative measure?
I'm not sure. I don't think that would help because it's not necessarily the same pathway we're interested in. So if you're getting genes that are not looking good at the end, I'm not sure if that relates to the actual, say, PXR pathway. That would be my response. I'm curious, Naresh, how you feel about that.
Yeah, I think Helen's talking about 18S, GAPDH.
Yeah.
I think a lot of companies do monitor those, but you're right. I mean, they're pretty abundant, right? They're not very sensitive to change because they're so abundant. But it's a good question, Helen. I think most companies look at morphology as well as housekeeping gene expression changes. And I believe when we curated the data within the working group, we looked at that as well. So Albert Lee again, are there clinical examples of bell-shaped curves?
So that's an interesting question. Does he mean that you see a decrease in fold induction clinically at a higher count? I would say no. I don't think. I would say this is actually an in vitro phenomenon, if I understand the question. I think the appearance of a curve that looks like this is solely because your system is unable to show induction anymore at these higher concentrations for whatever reason. But it's a good one though, right? If it's not, right? If it's not cytotox, it's not some screwed up mechanism, then you would see it in vivo, right? It's an actual phenomenon that is describable. I don't have any reason to think it is though. I think that's what he's asking.
Yeah, I guess also in the clinic, we generally don't do multiple doses, right? Mm-hmm. Like in vitro, we can do 10 concentrations. Obviously in the clinic, we can't do as many doses. So that's another reason why we may not be seeing it. But you're right, it could just be an in vitro phenomena. Yeah. Julius and Noru had a question as well. Julius says, very nice presentation. Can you comment on the use of GAPDH? Oh, I think we covered that one. Question from Dave. What are your thoughts on using LC-MS protein quant to evaluate induction? That's Dave Stress's question.
That's a good one. Is a science evolved where you can really detect that at sensitivity level that could help us out? Maybe, right? That's a good question. You'd also have the qualifier that the presence of the protein is means something. Right? I'm referring to inhibitors, right? You have to be careful there. But I think that's a good perhaps avenue to answer my philosophical question, right? If we could increase our sensitivity, we can get better initial slopes. So
yeah,
yeah,
it becomes interesting when you have TDI and induction, right? And you have this disconnect between mRNA and activity. So having that tiebreaker is a big plus.
yeah.
Question from Jason. I guess we'll take. We're at time, so we're gonna stop here. And questions that we didn't answer, Simon will follow up with those individuals and respond to their questions. So thanks again, Simon. Fantastic presentation. And thank you to the audience for attending.
Yes, thank you very much. And yeah, you can shoot me an email, and then yes, these slides will be available. Don't worry. But thanks again for your attention.